The overall goal of this direct thrombus imaging technique is to quantitatively visualize fluorescently labeled cerebral thromboemboli in vivo and ex vivo using gold nanoparticles, computed tomography, and near-infrared fluorescent imaging.
This method can help answer key questions in the hemostasis and thrombosis field, such as time or dose-dependent and serial therapeutic response of cerebral thromboemboli to tissue plasminogen activator or new thrombolytic agents under investigation. The main advantage of this technique is, after intravenous injection of gold nanoparticles, high-resolution microCT enables capturing dynamic evolution of cerebral thromboemboli by visualizing their locations and burden quantitatively in vivo, then corroborated by near-infrared fluorescent thrombus imaging ex vivo. Demonstrating the procedure will be done by Jeong-Yeon Kim, who is a postdoc from my laboratory.
After collecting 300 to 1,000 microliters of arterial blood by cardiac puncture, add 70 microliters of the sample to 30 microliters of the appropriate activated coagulation factor, 13-mediated fibrin cross-linking activity-sensitive fluorescent probe, and load a 3 milliliter syringe equipped with a 23-gauge needle with the resulting mixture. Next, inject the labeled blood into a 20 centimeter long polyethylene tube and allow the blood to clot at room temperature. After 2 hours,